recall these major concepts when we describe the action of the hormones, and they are as the following. Some hormones are synthesized in their active form and secreted immediately upon the action or upon the response or upon the triggering of the cell in order to produce these hormones. So some of them already active, already synthesized in the cell and just waiting certain trigger or certain signal in order to be released. While some other hormones synthesized in final form and stored in the producing cells until we receive another signal from the another kind of tissue. So these are, or these two types of hormones sharing the activity. So they are already formed, active, and waiting the signal, but some of them are secreted immediately and some of them waiting signaling molecules in order to be reduced. While the other types are synthesized as precursor molecules, it means that they are not active yet. They are just precursor molecules, and then they are processed and secreted upon physiological action or physiological activation or physiological need. So they are synthesized in active form and waiting the signal in order to be processed and then released to the cell. While some other hormones converted to the active form upon the reach or after reaching the target cell. So they need further activation at the site of the action. So these are the major types of actions that we could see when we talk about the activation of the hormones. Going back to the title of this lecture today, which is the steroidogenesis. Steroidogenesis as a concept means that you are going to synthesize steroids or steroid hormones from the cholesterol. So many hormones are synthesized from this precursor, which is the cholesterol, by modification of this molecule, and this family of hormones are called steroids or steroid molecules, and the conversion of cholesterol to these steroid hormones is called steroidogenesis or formation of steroids. We'll start with what's called adrenal steroidogenesis because we have other sites or other tissues that are responsible for the synthesis of steroids or steroidogenesis, which is the gonadal steroidogenesis. So we have adrenal steroidogenesis as well as gonadal steroidogenesis, which means that the testes or and ovaries are responsible for the steroidogenesis because they have similar enzymes that are responsible for the modification and metabolism of these steroid hormones. So basically, as we mentioned before, steroid hormones are synthesized from this precursor, which is the cholesterol, and the cholesterol itself is synthesized from acetyl-CoA, which it can be generated from either degradation of fatty acids or metabolism of the glucose, which is the process of oxidation of glucose, as you may remember. Now, in order to start this process of steroidogenesis by the adrenal gland, and you have to remember the anatomy of the adrenal gland because the different sites or tissues inside the adrenal gland are responsible for the synthesis of different types of steroid hormones. Generally speaking, the ACTH, adrenocorticotropic hormone, is responsible or is required in order to stimulate the adrenal gland. And I think we discussed this in brief, how the ACTH is going to activate the target cell. So the target cell in this situation is the adrenal gland, and there is a need for the esterase activity. Esterase, it means that you are going to release the fatty acid from the cholesterol molecule in order to generate the naked, if you would, or free cholesterol that is going to be processed to synthesize other precursors. Okay, and we'll see this in the next slide. The most important thing to remember when we talk about steroidogenesis is the following statement. It is shared between, I mean, as a location or as the site of the enzymes that are responsible for the steroidogenesis. They are either mitochondrial enzymes or endoplasmic, I mean, smooth endoplasmic reticulum enzymes. So The mitochondria as well as the smooth endoplasmic reticulum membrane, both of them are responsible for the metabolism of the cholesterol to synthesize steroid hormones, or briefly, responsible for steroidogenesis. Because there is a complex that is required in the mitochondria, which is called cytochrome P450. And this complex, that's why I'm saying complex, because it contains what's called SCC, if you remember, which means side chain cleavage enzyme that is localized in this complex, which converts the glucose to a pregnenolone. Pregnenolone is the other precursor, if you would, or the main molecule that is going to be converted to the other types of steroid hormones. Okay, they consider this molecule as a mother molecule for other kinds or other types of steroid molecules. In general, this process also requires what's called ACTH, adrenocorticotropic hormone dependent steroidogenic acute regulatory protein, okay, which is called STAR. This protein is essential for the transportation of cholesterol to the mitochondrial cytochrome complex in order to be processed. So this process of transportation of the cholesterol requiring the action of this hormone, which is, or I mean the action of this protein, which is depending on the ACTH hormone. So in order to make it easy, this is the cholesterol molecule that we are going to process. And as you can see, there are 4 fused rings from A to D. And this is the hydroxyl group that is playing an important role in the function of the cholesterol and the structure of cholesterol as well, and can be esterified in order to have cholesterol ester. So the esterase is responsible for the removing of the the fatty acid from the cholesterol molecule. The most important thing in the metabolism of the steroidogenesis is this hydrocarbon chain that is linked to the cholesterol molecule. And as you can see here, the first step in the formation of other types of steroids is the cleavage of this side chain, if you would, or this hydrocarbon chain in order to generate pregnenolone. So that's why we are saying that pregnenolone is the second most important molecule actually in the metabolism of the steroid molecules or steroids in general. So this cleavage is catalyzed by P450 or CYP or CYP, if you would, side chain cleavage enzyme that is localized in this complex, and it's activated by the adrenocorticotropic hormone through the action of the cyclic AMP as a second messenger. So the side chain cleavage again is going to generate this major precursor for other steroids. Okay, so the pregnenolone is the most important molecule that we are going to talk about today, and there are sites of modifications that will convert this molecule pregnenolone to other types of steroids. Okay, the other molecule, which is the side chain, is going to be processed separately, and we are not interested in the metabolism of this side chain group. So basically, in order to understand what we are going to talk about, we have to imagine what are these molecules that we are going to synthesize or to form. First of all, the estradiol molecule is one of the most important estrogens, as you might know, as well as the testosterone, which is an androgen, and glucocorticoid hormones like the cortisol, as well as the progesterone as a family of progesterone molecule. So these are the types of the steroid molecules that we are going to talk about today and how they are going to be synthesized by mitochondrial and endoplasmic enzymes. Okay, not by mitochondria itself, but by the enzymes that are localized in these organelles. So again, as I may describe before, we have to remember the structure of the adrenal gland that consists of these layers, and we are interested in the zona reticularis, as well as zona fasciculata and zona glomerulosa. Okay, because these are the sites or the tissues that are responsible for the metabolism of our steroid hormones as we see here. Okay, that's why the first title is called adrenal steroidogenesis, because we are going to describe the metabolism inside the adrenal gland, as you may notice here. So basically, we need these kinds of enzymes, and I am not very interested, or I don't want you to memorize every specific enzyme in these pathways. Okay, لانه انا بعتقد انه لا جدوى انه الان تحفظهم وبعدين تنساهم. I need you to know the basic concept of metabolism in each of these zones, okay, or these layers or these tissues in the adrenal gland, as well as the major processes of this, the metabolic pathways, like the hydroxylases that require the presence of oxygen and NADPH as a reducing agent, and dehydrogenases, isomerases, as well as Lyase enzymes that are required for the metabolism. So these are the major things that we are going to see, or the things that are required for the metabolism of steroid hormones in the adrenal gland. Starting with the first step, which is shared in all layers, as we described before, CYP SCC enzyme, اللي هو side chain cleavage enzyme, okay, that is responsible for the generation of a pregnenolone, which is the precursor, can be considered as a precursor for the steroidogenesis. Okay, so here we can see the specificity of the zona fasciculata that is responsible for the synthesis of glucocorticoids, which are the cortisol and corticosterone. Okay, and the process, as you can see here, starts with the cytochrome action, and we can see here the action of isomerase enzyme and isomerases, much more. And we can see here certain numbers. These numbers means that we have hydroxylation process or oxidation process at the carbon number. So these numbers mentioning the location of hydroxylation or isomerization or the modification of that or of these molecules. The most important thing here is to notice the following. If you take a look to the site of the previous side chain after the cleavage, you can see this portion or these arms in the glucocorticoid hormones as well as the aldosterone, but it's not in the androgens or estrogens. Okay, so there is kind of modification, major modification, to make these hormones belong to different families. That's why we are classifying the metabolism close to each other depending on the similarity of the molecules. So zona fasciculata sharing the zona granulosa in the synthesis of similar structures and generation of the glucocorticoid hormones as well as mineralocorticoid hormones. This hormone belongs to the mineralocorticoid hormone, and the others are called glucocorticoids. And I think you are familiar with this. On the other hand, the zona reticularis is responsible for the generation of these estrogens that will be converted to either androstenedione molecule, which is another kind of estrogen, as well as the active form of the DHEA hormone, which is known as dihydroepiandrosterone hormone, the active form or the active estrogen hormone. Okay, dissecting the metabolic pathway that we described before in order to know the similarities and the sharing pathways between these molecules in order to form either glucocorticoid or mineralocorticoid hormones or steroid hormones. So basically we start with the cholesterol again, and we have this major step, which is the cleavage of the side chain in order to generate a pregnenolone. So we have two options or two pathways, either hydroxylation at position number 17, for example, in this case, and that's why it's called 17 hydroxypregnenolone, and generation of dihydroepiandrosterone, and then androstenedione hormone. So these steps, as you can see here, are generated by a complex which is the CYP 450, that has hydroxylase activity as well as lyase activity in order to remove this or the rest of the side chain. And this is the difference between these two molecules. Okay. Or the other direction is the generation of the progesterone, which is another kind of molecule that can be converted to 17-hydroxyprogesterone, which is similar to the 17-hydroxypregnenolone, in order to generate the similar molecule at the end of the day. So there are two kinds of actions or two pathways that can be followed in order to generate the same molecule. Or these two molecules can be generated or can be converted by hydroxylase enzyme at position 21 in order to generate at the end of the day corticosterone or cortisol. The point is to know that we have sharing pathways that, or in fact, two kind of options in order to synthesize these hormones depending on the activity of these complexes and the availability of these enzymes. So hydroxylases, lyases, and oxidase enzymes are required for the synthesis of these hormones, and these enzymes are localized in these complexes, which can be at the beginning located in the mitochondria, and the rest are located on the endoplasmic reticulum, and specifically it's the smooth endoplasmic reticulum. The corticosterone hormone can be converted, the last one, from glucocorticoid hormone to the mineralocorticoid hormone by this last step at the position of zona granulosa by hydroxylation in order to form what's called aldosterone. هلا الالدوستيرون الفانكشن تبعه مختلف تماما، عشان هيك سمينا mineralocorticoid hormone, which is responsible for the regulation of the water released by the renal system. هلا mineralocorticoid again, starting from the beginning, starts with the cholesterol, pregnenolone, and then we have hydroxylation steps, and finally we have aldosterone synthase enzyme that is going to convert the glucocorticoid to the mineralocorticoid hormone. And this is just a revision of what we described before. هلا last step اللي شفناها هون هي نفس الستيب اللي هون, and these are the other steps. فأنا شلتهم من هون وحطيتهم في خطوات متتالية in order to understand the mechanism of the generation of the aldosterone or generation of mineralocorticoid from the glucocorticoid. Going back to the glucocorticoid hormones, these are the glucocorticoid hormones that we described before, and they have kind of similar structures except the branching between these types of glucocorticoids. And the process of glucocorticoid synthesis is performed in the zona vesiculata and zona reticularis. كل طبقة منهم specified or has special kind of enzymes that are responsible for the production of certain hormone. This is very important. Very important thing to remember is the final step of the generation of the cortisol is performed by the mitochondrial site, while the previous steps are by smooth endoplasmic reticulum. And here at the beginning, we have to remember this is performed by the mitochondrial enzymes as well. So we start with the mitochondria and end up with the mitochondria. In the middle, we have the smooth endoplasmic reticulum enzymes in order to generate our target hormones. On the other hand, we have androgen hormone, which is, as I mentioned before, is different from the glucocorticoid or mineralocorticoid hormones by having the carbonyl group at this position in order to distinguish it as a androgen hormone that is synthesized by the adrenal gland, اللي هو واحد من active hormones as a DHEA hormone, and it will be activated, as I mentioned before, by sulfotransferase by having sulfur group. Now, what we described before is related to the adrenal gland, adrenal steroidogenesis, and here we are going to move to the gonadal or gonad steroidogenesis, and the first gonadal system is the testicular cells. The testicular cells are responsible for the formation of the androgens, and the specific cells that are responsible for these actions is the Leydig cells.
the conversion of cholesterol to the other to the next step active or to the next molecule as another precursor in the steroidogenesis, which is the pregnenolone, is identical in the testis and ovary and in the adrenal gland, while in the ovary it needs luteinizing hormone, which is the LH, while in the adrenal gland needs the ACTH hormone as an activating hormone. The process requires the action of group of enzymes, and there are 5 enzymes that are contained in these proteins. What does that mean? It means that you have 3 proteins and they have 5 active sites, 5 active enzymes, or if you would, domains that are responsible for the generation of these androgen hormones. And the major androgen that we are interested in is the testosterone molecule. So we have these enzymes, as you can see, making a complex structure in order to generate or to convert the pregnenolone by either this pathway, which is called dihydroepiandrosterone pathway, or delta 5 pathway, or delta 4 pathway, which is called the progesterone pathway. So we have this direction of the testosterone synthesis, or the other direction of the testosterone synthesis in the testis. Okay, eventually you will have the inactive or less potent form as androgen hormone, which is the testosterone, which means that we need further step in order to activate this hormone in order to be effective. So going back to the process of the metabolism, we need at the beginning hydroxylation in either direction, whether you are going to start with the pregnenolone and generation of 17 alpha hydroxypregnenolone, or generation of progesterone, and then the progesterone is going to be converted to 17 hydroxyprogesterone. So we have eventually 2 similar molecules, I mean, from the point of view of the hydroxylation or the position of the hydroxylation. So generation of this molecule is going to be converted to the other one, while the generation of the other molecule by hydroxylase is going to convert eventually the other molecule. So we have kind of interactions in each step because we have this enzyme, which is the isomerase enzyme, that is going to convert the pregnenolone precursor to the progesterone precursor, and eventually we will generate the testosterone hormone. Okay. Few seconds earlier, I mentioned that this hormone, which is the testosterone, is less potent. What does that mean? إيش يعني less potent؟ يعني الactivity تبعته as a hormone, as an androgen, is not highly effective. So we need further step of activation or generation, the highly potent testosterone or the more active testosterone hormone. And this process requires the presence of this enzyme, which converts the testosterone to the dihydrotestosterone, which is the active or highly potent molecule. This enzyme is 5-alpha reductase enzyme. So we have a reduction process at this position. Okay. And this is airing, as you might know, that is going to be reduced. And the reduction process requires the presence of NADPH molecule. Okay. And this activation can be performed in the testis, I mean the source cells, or in the peripheral cells, اللي هو target cells. So the testosterone is going to be metabolized. In fact, it's going to be activated by conversion of the testosterone to the dihydroxytestosterone, or sorry, dihydrotestosterone. And this process is activated by the reductase enzyme. Okay. So we have oxidation at position 17 in many tissues, as we mentioned before, to produce less active ketosteroids, and we are not interested in this. It's the process of metabolism of the testosterone and the reduction of the airing, which we mentioned before, double bond, and the 3-ketone position in the target tissue to produce the potent metabolite اللي هو dihydrotestosterone molecule. So, as I mentioned before, it can be generated in the testes themselves, or can be triggered or generated or acted by the target cells after reaching the target cell, if the testosterone is going to reach that target cell. So this is the major metabolic actions that will face the testosterone after the synthesis of this hormone in the target tissue. I mean the source tissue when I say the target tissue of the testosterone. The other site that is responsible for the steroidogenesis is the female organ or gonadal system, اللي هو ovarian steroidogenesis process. So we have a group of estrogens, اللي هم group of female hormones إنجاز التعبير. And the 17 beta estradiol is the primary estrogen of the ovarian origin. It's one of the most important hormones that is recognizing estrogens or ovarian steroidogenesis process. Theca cells are the source of the androstenedione and testosterone that will be processed in order to form the estrogens. So we have to have androgens at the beginning in order to form estrogens in the female organs or in the ovarian gonadal system. So again, we have androgens that are going to be converted to estrogens. So we need a specific mechanism that is available in the ovarian tissue in order to generate these estrogens, which is not available in the testes, for example, in order to form estrogens. However, we can form certain kind of estrogens for certain extent. Granulosa cells also responsible for the conversion of androstenedione and testosterone to estrone and estradiol. Estradiol is the active form of the female hormones, or estrogens, while estrone is the less active form of the hormone. Talking about the mechanism of the metabolism or generation or conversion of the androgen to estrogen, we have to recall what we learned before. We start with cholesterol and side chain cleavage enzyme is responsible for the generation of pregnenolone, and we have two pathways of the androgens that we learned before, اللي هو hydroxypregnenolone or hydroxyprogesterone, in order to reach the final molecule, which is the testosterone as an androgen. Because we have aromatase enzyme that is available in the ovary, we will generate the estradiol, 17 beta estradiol molecule, which is the potent molecule, and this is the less potent molecule as an estrogen, which can be generated either from the estradiol or from the androstenedione hormone by the aromatase enzyme that is available in the cytochrome complex system. So as we notice in the cytochrome, P450 is the most important complex that is responsible for the metabolism of steroid hormones or the steroidogenesis processes. Okay, so we can generate the active form of estradiol from the androgen, as I mentioned before, and we can also convert the estrone to estriol, which are the other metabolites with less potent action as steroid hormones. In the corpus luteum, we have this mechanism of the progesterone synthesis from the same precursor molecule, and this means that the cholesterol is going to be synthesized from the degradation of the degradation fatty acids and then generation of acetyl-CoA, or from the metabolism of glucose. So these are the most important mechanisms that you have to remember when we talk about the steroidogenesis process, the adrenal and the gonadal steroidogenesis mechanisms. Some few points that you have to remember when we talk about the gonadal hormones or steroid hormones, if you would, which means that there are kind of significant amounts of estrogens are produced by the peripheral aromatization of androgens. What does that mean? It means that, as I started my lecture, when we talked about the precursors of hormones that will be released upon the stimulation, but still they are precursor and they need to be activated at the site of action, as we can see here, there is a peripheral aromatization of androgens, which means that there are certain kind of tissues can convert androgens to estrogens rather than the gonadal system itself. So the peripheral tissue is responsible for the aromatization process, and it depends on the gender. For example, in males, the peripheral aromatization of testosterone to estradiol accounts for 80% of the production of the estradiol. Okay, so the estradiol is going to be activated, or most of the estradiol that will be generated in the male organs, or in the male, I mean, is the responsibility of the peripheral tissues, okay, not the testes themselves. While in females, 50% of the estradiol produced during pregnancy comes from the aromatization of the androgens. Okay, so there's kind of different in the percentage of aromatization depending on the gender itself. So in either direction, we have peripheral aromatization or peripheral activation of the precursor molecule. So in this case, the precursor is the testosterone, while the estradiol is the end product or the target molecule that we want to synthesize. The last but not the least, which is another kind of debating molecule as a hormone or as a vitamin, which is the vitamin D. Okay, so vitamin D is considered as a vitamin, but in fact, it's a precursor of steroid molecules. So it is generated from the cholesterol, and it's a derivative of the cholesterol molecule. And there are kind of certain chemical reactions, or in fact, there are pathways in order to synthesize the steroid hormone in its active form. And as you may remember, we have to have sunlight in order to activate what's called conversion of the dihydrocholesterol molecule to pre-vitamin D3 structure. And this process is performed or conducted in the skin cells. And this process is non-enzymatic. That's why it's called non-enzymatic photolysis reaction. It's kind of photolytic action that is required in order to generate the pre-vitamin molecule as the, if you would, or if you remember the pregnenolone molecule as another precursor, here we are, here we have what's called pre-vitamin D3. So in order to be converted in the liver or metabolized in the liver by certain enzymes in order to generate vitamin D3, okay? And it's transported by vitamin D binding protein in order to be transported from the skin or from the intestinal side to the liver. Why we need such kind of transporter? Because this vitamin is water insoluble. So we need, or we have to have a carrier for this vitamin or this molecule. So next, by hydroxylation, you will generate further hydroxylated molecules at different positions in the kidney, either at position 24, 25, or 1 and 25 hydroxylation, and you will generate this metabolite or derivative molecule in order to have in the future these structures. And this is the active form that we need in order to have the action of this vitamin. Okay. So the kidney, skin, liver, and kidney are responsible for the metabolism of vitamin D in order to have the active form and other metabolites with different potent activity of this vitamin. And this is the last thing about the steroidogenesis process. So the next thing that we have to describe today is the catecholamine and thyroid hormone synthesis. Let me have a small break here, like 2 minutes, and then we'll go back. I'll stop recording. اوكي كلنا سامعين بعض. اذا في حدا اوكي رنيم.
دكتور بدي أسأل سؤالين بالمحاضرة اللي أخذناها عن قبل شوي إذا في مجال.
عارف سؤالين. الانسولين افكت رايت؟
لا بدي أسأل باللي أخذناه قبل شوي، بس إذا في مجال دكتور تفتح السلايد.
تمام. معناته خليني ارجع اعمل ريكوردينج. اوكي، او خليني اشوف. يس. اوكي.
بسلايد 6
يس.
لهون اللايز انزيم بيشتغل على الموقعين 27 بنفس الوقت، على الكربون 27 في نفس الوقت، ولا يعني كل مرة بختار كربون مثلا؟
بعد شوي. اوكي. it's carbon 21.
تمام. سؤالي إنه هون اللايز انزيم بيشتغل مرة على الكربون 7 ومرة على الكربون 20، ولا بيشتغل على الكربونتين مع بعض في نفس
لا لا
الوقت؟
لا هو الهيدروكسيليز enzyme is different from each other. هذا كل واحد مختلف عن الآخر. Okay, we have 21 hydroxylase and 11 hydroxylase, for example, in this case.
دكتور بسأل أنا عن اللايز مش الهيدروكسيليز.
لايز. أنا قرأتها غلط. سوري. hydroxylase. وين أنت لايز؟ أنت غلطتيني. هون. it's the same enzyme here. It's the same molecule, the same enzyme.
هلا هون مكتوب عندنا 27، يعني قصدي هون مرة بيشتغل على الكربون 7 ومرة 20،
بنعمل
ولا عليهم على الكربون في نفس الوقت؟
بنعمل لايز activity على two positions هذول.
at the same time.
لا هي sequential.
يعني بيشتغل.
it's lyase enzyme. It has to cleave stepwise.
تمام. دكتور سؤالي الثاني في سلايد 17.
تمام. 17 هون.
هلا تمام. احنا حكينا الاندروجين بيعطينا تستوستيرون، صح؟
يس.
تمام. ممكن احكي ان هذول التفاعل يعني الرياكشنز اللي بتصير هون تو دايركشنال، يعني تفاعل باتجاهين هون بصير، كونه مرة بتحول الاستيرودول على تستوستيرون وبالعكس.
لا بصيرش الاستراديول بتحولش لتستوستيرون. تمام. الاتجاه واحد، هو من التستوستيرون للاستراديول. بمعنى اخر you have to remember this انه الاندروجينز generate الاستروجينز،
اما العكس ما بصير.
لا لانه في الباثوي if you remember here. Where is it? Okay, so you start with the assuming you started with the cholesterol and pregnenolone, and either pathways will generate the testosterone. So there is no more reactions that, or there are no reactions that will generate testosterone rather than the steroidogenesis pathway اللي هي الميجر pathway. هلا هون خليني بس اشوف، انا بتذكر فيهم السلايد I have shown in. Okay. No, it's androgen. Okay, so just remember that the androgens are converted to the estrogen. Yeah, this one, as you can see here. Okay, so generation of androgens can be considered as a precursor for the generation of the estrogens. Okay, and these are the enzymes, or this is enzyme that actually the enzyme that is responsible for the conversion of the androgens to the estrogens. It's the aromatization or aromatase action. تمام. هل انا بعتقد انه كان سؤالك، كنت مفكر يعني سؤالك متعلق باللي يسالون عن الطلاب and I'm very thankful انه سالوه بهذاك الوقت. كان بيتعلق بشيء خاص بالانسولين action, and let me go back to the lecture in order to clarify it. There was something wrong in the text itself. One of them was wrong from the biochemical point of view. Okay, which is this one. So we have this slide, and we have, okay, the other one. Where is it? Okay, anyways, this slide says that the insulin is responsible for the activation of glucose synthesis. Actually, it's the glycolysis process, not the glucose synthesis. Okay, so this is not right. Okay, and I think we have another slide for the same diagram. Yeah, I think you have to change it. It's glycogen synthesis, not glucose synthesis, this one. Okay? Because the gluconeogenesis is deactivated by insulin, while glycolysis is activated. Or if you would, glycogen synthesis is activated. So it's glycogen, not glucose. So let me correct it here. Okay, I will interfere with the slides. Okay. To make it appropriate. So it's okay. Save it. So this is the correct one. Glycogen synthesis, not glucose synthesis.
دكتور
Yes. ارفعي صوتك.
هسه الاستراديول هو يعتبر الاكتف فور بوث التستوستيرون والاستروجين صح؟
لا نقدرش نعتبره هيك. هلا هو مش اندروجين، صار استروجين. فهو another active form as a different molecule as estrogen rather than being androgen. Yes.
يعني هو مش
اذا بتقصدي فهمت عليك انه التستوستيرون راح يتحول لدايهيدروتستوستيرون ويتحول للاستراديول. هلا دايهيدروتستوستيرون is the male hormone, the active one or highly potent one. عشان هيك انا بستخدم كلمه less potent و high potent. التستوستيرون له اكشن بس الاكشن very minimal. يعني اذا بتقارني تقريبا حسب الليترشر من 25 ل 100 ضعف كاكتيفيتي of the dihydrotestosterone compared to the testosterone itself. Fine. So still it has an action, but it's very weak. تمام. هلا الاستراديول is another molecule. It's not the modified testosterone. It's estradiol molecule which is working in the in females. تمام. إذا مش واضحة احكي لي. لأنه هذا بصير يعني اثنين بصيروا two organs by I mean two organisms but the concentration is different from one organism to another. Any other questions?
دكتور عملية الأروماتيزيشن هي بتصير بس بالأنثى ولا كمان يعني صح هو بس للفيميل مش للميل صح؟
Yeah, mainly in the females.
طيب ليش حكينا لما عن النسب حكينا إنه 80% الأروماتيزيشن؟
حكيت لك كلمة mainly. So we have estradiol in males. بس البورشن تبعهم yes. البورشن تبع الاروماتيزيشن بغض النظر عن الكميه. هلا الكميه راح تكون اقل بكثير من الفيميل. تمام. والاروماتيزيشن بصير بالperipheral tissues for certain actions for certain mechanisms or stimulations. But البورشن تبع هذا السمول concentration is 80%. كلمه 80% لا توحي لكم انه it's very huge amount. يعني ممكن تكون على سبيل المثال للمقارنه as an arbitrary unit لو عندنا 100 يونت بالفيميل تمام 50% منهم راح يصير aromatization بالبريفرالز. لو عندنا يونت واحده بالميل لاحظي انه الفرق يونت واحده و100 بالفيميل. يونت واحده بالميل 80% من هاي اليونت will be aromatized in the peripheral tissues. هلا الاروماتيزيشن ككميه لا تعني انه هذا very or present in high concentration in males. Thank you for the asking. يعني هو مش major one in male.
تمام شكرا.
لأنه استروجين بالنهاية. طيب بما إنه الوقت almost is going to be finished within 4 or 3 minutes. خلينا نأجل هذا للمحاضرة الجاي اللي هي بعد بكرة إن شاء الله الساعة 11 إذا أنا مش غلطان حسب التايم تيبل اللي عندي. طيب إذا في أسئلة وإذا ما في نقدر الآن نغادر المحاضرة نكمل المرة الجاي. إحنا we have time يعني we are managing the time very well. Okay. Thank you very much.